the GAL4 UAS system in Drosophila, produced by Todd Bonesteel, Evan Reese, and Joseph Tredella for Molecular Biology of Model Systems Markstein Module, Spring 2016, University of Massachusetts Amherst. What is GAL4 UAS? The GAL4 UAS system is a technique for expressing transgenes in specific cells and is employed in a variety of organisms. This presentation will first give some background on how genes are regulated and then go into detail on how the GAL4 UAS system works and why it is important. So what does it mean for a gene to be turned on? A gene being turned on means activating transcription of that gene to mRNA, which is often followed by translation into a protein. Various proteins are used in a variety of ways in virtually every cellular process. Every cell possesses a complete copy of the organism's genome, and so could theoretically express every gene in that genome. This point was proven by the classic frog cloning experiment. The frog cloning experiment was conducted in the 1970s by a researcher named John Gurdon. He destroyed the nucleus in an egg from frog A using UV radiation, and replaced it with the nucleus from a differentiated skin cell from frog B. The egg was then able to develop into a tadpole genetically identical to the donor frog, frog B. This proved that the skin cell nucleus still contained the full frog genome. However, all genes are not expressed in all cells, nor are they expressed at all times. We can understand this by considering the fact that our eyes aren't expressing the genes that make hair grow from our scalp. Also, the genes that control our hair color change over time, which we can see as hair tends to gray with age. Some genes are also regulated over much shorter time spans, such as those coding for proteins involved in circadian rhythms like melatonin, a hormone that makes us tired. Regulation of genes is complex and involves various elements including transcription factors, which bind to specific sites on the genome to control the rate of transcription. One category of transcription factors is activator proteins, which bind to enhancer sequences on DNA and trigger transcription by recruiting RNA polymerase to bind to the promoter sequence. The GAL4 UAS system exploits this molecular mechanism to drive gene expression in a specific way. GAL4 UAS was developed in 1993 by Andrea Brand and Norbert Paraman at Harvard Medical School. The system consists of two separate parts, the GAL4 gene from yeast, which encodes the transcription activator GAL4, and the upstream activation sequence, or UAS, an enhancer that the GAL4 protein binds to in order to activate transcription of an associated responder. GAL4 UAS is particularly useful for artificially controlling gene expression in Drosophila because GAL4 is not normally present, and so its expression does not interfere with other processes in the cell. The GAL4 gene itself must be turned on and off, and so for use in Drosophila, it is placed under the control of a native gene promoter or driver. The separate UAS sequence is fused to a target gene of interest, known as an effector, responder, or reporter. Since GAL4 is only expressed in cells where the driver is active, the responder will also only be activated in those cells. The UAS responder is present in all cells of the animal, but in most cells nothing happens since GAL4 is not present to activate it. In the cells that are expressing GAL4, however, the UAS is activated, the reporter gene is transcribed, and the reporter protein is produced. A common reporter is green fluorescent protein, or GFP, which glows green in response to blue light. These images show UAS GFP being expressed in the central nervous system of a fly larva and in the antenna, proboscis, and at the base of bristles in an adult fly's head. In practice, flies carrying the UAS responder sequence, known as reporter lines, are crossed with those carrying the GAL4 driver to create a fly that will express the gene of interest in a particular way. It does not matter which chromosomes GAL4 and UAS are on because translation from mRNA to protein occurs outside of the nucleus. After translation, the GAL4 protein enters the nucleus and can bind to the UAS region on any chromosome and activate transcription of the reporter. So, why bother with separating the GAL4 and UAS components of this system? Isn't that just creating more work? Let's say we wanted to study the interaction of 4 enhancers with 4 responders. We would have to create 16 direct fusions by embryonic injection. This is a time-consuming and expensive process that also requires knowledge of the enhancer being studied. With GAL4 UAS, you need only to cross existing GAL4 enhancer and UAS lines to create the flies for the study. The flies do the work, saving researchers time and money. The advantage of GAL4 UAS over direct fusion becomes very clear when we consider the level of cooperation that goes on in the fly research community and the costs associated with creating direct fusions. Many researchers choose to share their unique GAL4 lines via fly libraries like the Bloomington Fly Stock and the Vienna Drosophila Resource Center. These two libraries alone contain over 15,000 GAL4 stocks of Drosophila, and there are also over 100,000 UAS responder lines available. At $200 per construct, the cost of creating direct fusions for every possible combination without GAL4 UAS would be around $310 billion. With GAL4 UAS, it is basically free to create these combinations. To summarize, the GAL4 UAS system is used to express transgenes in specific cells across a variety of organisms. The GAL4 protein is a transcription factor known as an activator. GAL4 binds to UAS, an enhancer, and activates an adjacent reporter gene such as GFP. Vast stocks of GAL4 and UAS responder lines in Drosophila allow researchers to mix and batch to create new lines with relative ease.